Good morning. My name is Ross Walenga. I'm a chemical engineer in the Division of Quantitative Methods and Modeling in the Office of Research and Standards. And this morning, it will be my pleasure to speak to you about bridging the gap between regional deposition and systemic pharmacokinetic data of OINDPs with modeling and simulation. I have several learning objectives for you this morning. I'd like for you to be able to identify the sites of action for various orally inhaled and nasal drug products, or OINDPs, to describe different in vitro and in vivo methods for regional deposition data collection, to determine strengths and limitations of various modeling techniques for prediction of regional deposition, and to be able to formulate strategies for evaluating differences in rate and extent of drug delivery to the site of action for OINDPs with modeling. So to begin the talk, I'd like to discuss what is written in the regulation for bioequivalence, or BE. In 21 CFR 314.3, it states, bioequivalence is the absence of a significant difference in the rate and extent to which the active ingredient or active moiety and pharmaceutical equivalents or pharmaceutical alternatives becomes available at the site of drug action when administered at the same molar dose under similar conditions in an appropriately designed study. So I've highlighted here the absence of a significant difference in the rate and extent and available at the site of drug action. So this morning, I'll be speaking about the site of drug action, how that's important, and how we can show that there is an absence of a significant difference in the rate and extent for locally acting OINDPs. So for BE at the site of action, for orally inhaled drug products, or OINDPs, lung tissue concentration is the site of action. If you look at this diagram on the right, for an OINDP, the drug would be inhaled, and then it's either going to go to the gastrointestinal tract, or GIT, or to the lung. And even if it goes to the lung, it can still be moved to the GIT via the mucociliary escalator. So anyway, any drug that makes it to the lung and then is absorbed is actually drug that's delivered to the site of action, so local lung tissue concentration. Nasal cavity tissue then is the site of action for nasal drug products. So talking about things that we can measure either in vitro or in vivo, we can measure regional deposition and we can measure systemic pharmacokinetics, or PK. However, regional deposition is upstream of local tissue concentration and systemic pharmacokinetics is downstream. So it can be quite difficult to actually measure the rate and extent of drug delivery to the site of action for these products. So via the pre-ANDA process, OGD has received several communications about alternative BE approaches for OINDPs. In more than one case, the alternative BE approach focused on systemic PK for OINDPs, but this can be problematic because it's not actually giving assurance that the rate and extent of delivery to local lung tissue PK is the same. Another approach that we've seen, and Dr. Hahn spoke about in an earlier talk this morning, is to infer regional deposition based on systemic PK, but without validating these regional deposition values. So this certainly is a stronger approach than looking at systemic PK because it is bridging the two areas, but there's no assurance that these regional deposition sort of predictions, you could call them, are actually correct. So an ideal approach may then be to validate both your regional deposition and systemic PK predictions on both sides, and then bridge these two components to credibly predict local tissue PK if you're going to use a model for your alternate BE approach. And why would you use a model? Well, for alternative BE, you may have several in vitro and in vivo studies, like we mentioned, but none of them are likely to actually measure tissue concentration at the site of action. So I would like to talk a little bit about small airways for OINDPs. So if we look at the lung, it can be divided into several regions, either by gas transport and gas exchange, conducting an acinar, or by large and small airways. Typically for these type of drug products, we talk about large and small airways just due to differences in say, mucus layer thickness, metabolism, things like that. There's different properties for the different parts of the lung. And there's a cutoff for this, but there's some disagreement in the literature about where this actually is. So this could either be after the bronchi or for airways smaller than 2 millimeters in diameter. And it may even be beyond the bronchioles if we're talking about 2 millimeter diameter, so that could be a problematic definition actually. So there's some disagreement about where this is, but it's generally agreed that there is a significant difference in the way that the lung behaves between the large and the small airways. For OINDPs that target asthma as the disease, this is typically thought of as a large and small airway disease. So looking at this figure on the right, this figure is showing different immunocytochemical cell markers for either A, small airways, or B, large airways. And looking at the differences between the asthmatic and the control subjects, you can see some significant differences in these markers for both these regions. So it indicates that asthma is active in both regions of the lung, large and small airways. So for locally acting OINDPs, we can have several different types. We can have bronchodilators or corticosteroids, and even among bronchodilators, this can be divided into beta-2 agonists or antimuscarinic agents. So there's several different types, but typically, no matter what type of drug product you've got, it'll be ideal if you can target both the large and the small airways. So for COPD, or chronic obstructive pulmonary disease, this is typically thought of as a small airway disease. And again, you can have the same types of drug products, but ideally your drug product is going to be targeting the small airways. And so why does this matter for our discussion this morning? So say you have a generic compared to a brand name drug product, and your brand name does a very good job of targeting the small airways, and you develop a generic that has the same total lung deposition, but doesn't do as good a job of targeting the small airways. Well, it may not be as effective as a drug product in that case. So when we're talking about B at the site of action, we really need to understand where does the drug need to go, not only just to the lung, but where in the lung. For nasal drug products, the common disease that these drug products treat is allergic rhinitis, where the site of action may be the inferior turbinate. And if you look at this picture on the right, that can be that's this little bony outcropping down here. Nasal polyps is another disease that can be targeted by nasal drug products, which can either be targeted at the middle meatus, which is this kind of circular region here, or the middle and superior turbinates, which are these two bony outcroppings here. I mentioned before that it's possible to measure regional deposition. So how do we do that? Dr. Bielski spoke about this in one of the talks earlier this morning for an in vitro method that uses a realistic mouth-throat model. So in this case, you have a wrapper prototype model, and then you subsequently use high-performance liquid chromatography to measure the deposition in the various section regions. And this can even include the tracheobronchial region, but it's typically limited to the large airways just due to issues with rapid prototyping and the resolution of rapid prototyping. It's also possible to do this with nasal models. It can be quite difficult to section these precisely, but it is possible, and there's certainly promise in this area as well. We can also measure regional deposition in vivo. This can be done using gamma scintigraphy, which is a two-dimensional image. And from this, we can get central to peripheral ratio for lung deposition. This can be somewhat problematic because there's no generally accepted method for determining where these central and peripheral regions start and end. And also, even if there were, it is a two-dimensional image, so there's naturally going to be some overlap of large and small airways. But it does provide in vivo data, so it's certainly useful. If we look at the image on the right, this is a gamma scintigraphy study, and it was comparing two drug products to show that one drug product targets the olfactory region more than the other drug product. Another method is single positron emission computed tomography, or SPECT, and may be combined with computed tomography, or CT, SPECT-CT. This can provide three-dimensional information, so this is certainly a promising technique as well. What about PK data? Well, we know about systemic PK. This can be used to get plasma concentration. But what about local tissue PK? So I mentioned before, it's maybe something that we don't have. It's certainly difficult to obtain due to ethical concerns. It is possible in some instances to get these data using perhaps something called bronchoalveolar lavage, where you do kind of washing of the lung. But typically this is only indicated for patients that actually need this procedure, where the physician needs to know more about what's going on in the patient's lung. So it is possible to do a study, but it would be certainly limited and probably not practical for BE comparison purposes. For nasal drug products, during nasal surgery, it's possible to obtain tissue concentration measurements as well. Okay, so we've talked about how do we measure regional deposition, how do we measure PK, but then we'd like to know how can we model it, because with a model we can potentially tie together our alternative BE approach, because maybe we've got several in vitro and in vivo studies. We want to show that these studies are indicating that our local tissue PK is the same. Semi-empirical regional deposition modeling is one type of model that may be used. This is an algebraic semi-empirical model. Produces branch-specific deposition probabilities, and then these probabilities are used to sum across the different branch levels to obtain a regional deposition. And it's been developed for toxicology, so there's some limitations there because it's then been adapted to an aerosol-based products. But it can be it's a potentially useful method and certainly computationally efficient, and with proper validation could be an important part of an alternative BE approach. Physiologically based pharmacokinetics, or PBPK modeling, is a compartmental model that allows for prediction of local and systemic PK via a couple different considerations: dissolution in mucus layer, lung tissue absorption, lung tissue metabolism, and then ultimately it will be integrated with a systemic model and then validated with in vivo PK model. So on the left you can see this was a study using a PBPK model and compared with in vivo data. And then for the model, there were two different methods for obtaining regional deposition predictions. One using the native solver and the software that was used, which was GastroPlus, or using a different model, which is a multiple path particle dosimetry model. So depending on the regional deposition predictions, it actually changed it somewhat. There's computational fluid dynamics, or CFD modeling. This allows for prediction of fluid and particle transport. It's a physics-based modeling approach. Allows for consideration of realistic geometries, which can be really important for a drug-device combination product. So you can actually consider differences in device design. This may be validated with either in vitro or in vivo data, as I'll discuss a little bit later. And just to give you a flavor for what this looks like, the video on the right is showing fluid transport in a meter dose inhaler. The video on the right at the bottom is showing particle transport in either an MDI or a dry powder inhaler. And from that, we can then kind of back out our regional deposition predictions. So I'd like to speak a little bit about a model that's combined several different approaches. This is a nasal absorption model using CFD. It's GDUFA-funded research done by Virginia Commonwealth University. And the purpose was to predict local delivery of nasal suspension spray droplets using CFD. As you see on the right, in part A, the deposition was predicted, and then it was transferred to a surface model, which then predicted mucociliary transit with dissolution and absorption, all simultaneously. From this concept, this was connected to a PK model, which included a nasal epithelial tissue compartment, so your local tissue PK, and also a systemic model. So in this case, it was validated against systemic PK data. using a Cmax value from literature. So maybe not the strongest validation, but it gave some assurance that this model platform could be a useful platform. And so as you can see, you can validate your PK, and then you could also go back and validate your regional deposition, and then you would have local tissue PK predictions. Okay, how do we know, though, if this model is believable? How do we believe this model? So one particular standard available is the American Society of Mechanical Engineers, or ASME, Verification and Validation 40. So this is nothing that's endorsed by FDA officially, but it's just the standard that you can look up. It's available, and you could use it and may be useful to you. So how this works is you take something called the context of use, which describes what question is your model answering, and how much is the model being used to answer that question. From there, you determine your model risk, and then that's dependent upon what's the consequence of a bad decision, how much you're using your model. So if you're using your model a lot to make a decision with a heavy consequence, that could be a high-risk model. And if you have a high-risk model, then you may need a lot of credibility activities, such as verification and validation, to show your model is believable. So without talking too much about verification, I'll talk about validation a little bit. So say you're trying to validate regional deposition predictions, for example, and I mentioned CFD before. So in this case, you could compare CFD regional deposition predictions to data collected in vitro with a rapid prototype model. Or you could compare it with in vivo data collected with gamma scintigraphy. So in the figure on the right, you can see CFD predictions compared to gamma scintigraphy data for both central and peripheral regions. So you're validating both your large and your small airway deposition. So for local lung tissue concentration, this is our target region. We would like to be able to have these kind of measurements. Typically, systemic PK is much more easily validated, but it may be possible to have some lung tissue PK in some instances. You may have animal data or even human BAL data. So if you're lucky enough to have either of those, with animal data, you may be able to use some allometric scaling, and with your human data, you could perhaps even validate directly. So this is probably not going to be a common case, but if you do have these data, you should certainly use these. So I have a challenge question for this talk. The site or sites of action for OINDPs that treat COPD is or are the A, small airways, B, large airways, C, large and small airways, or D, nasal cavity. And the answer is A, small airways. So what are some conclusions from this morning's talk? The rate and extent of delivery to the site of action for locally acting OINDPs is best reflected by your local tissue concentration. Okay, great, but we know that there's problems with measuring that, right? So what we can do is then use a model, either regional deposition, CFD, or PBPK, in various combinations to predict local tissue concentration. And then from there, we can validate these predictions using deposition and systemic PK data when we don't have these kind of local lung tissue data. Of course, if we have those, we'll certainly use those to validate our model as well. But if we validate on both sides, that can produce a credible model that could be a useful component of an alternative BE approach. I'd also like to reiterate that if we're talking about lung tissue concentration, the site of action may be in the small and or large airways. So we certainly need to consider that in our model as well. I have some acknowledgements for the various people in my office who have contributed to this work. And now I'm ready for your questions.